Good day. My name is Jody Smith, and this is my Bioinformatics presentation for General Biology Honors. The gene that I chose for my presentation is the chromodomain helicase DNA binding protein 8, also known as ATP-dependent helicase CHD8. Other gene names that it most commonly goes by is CHD8. Taxonomy, of course, is Homo sapien, and the subcellular location is located within the nucleus. The chromosomal location for the CHD8 gene is located on chromosome 14 at 11.2. The CHD8 gene provides instructions for making proteins that regulate gene activity through a process known as chromatin remodeling. Chromatin is the complex of DNA and protein that packages DNA into chromosomes. The structure of chromatin can be remodeled to alter how tightly the DNA is packaged. The CHD8 protein is thought to affect the expression of many other genes that are involved in fetal brain development. Specifically, CHD8 protein and the gene it regulates is thought to help control the development of neural progenitor cells, which give life to nerve cells, and the growth, division, and maturation of neurons. In layman's terms, the CHD8 protein helps to control the number of neurons in the brain and prevents overgrowth. People with a missing or mutated copy of CHD8 often have autism, intellectual disabilities, or macrocephaly. CHD8, which encodes chromodomain helicase DNA binding protein 8, is one of the few well-established autism spectrum disorder genes. Autism spectrum disorder, or ASD, is a neurological and developmental disorder that affects how people interact with others, communicate, learn, or behave. Currently, over 60 autism gene mutations have been reported in subjects with variable phenotypes. The natural variant describes the naturally occurring differences in DNA sequences that are found among individuals of the same species, and such genetic dissimilarities arise from random mutations, which may be passed down to offspring. With the CHD8 gene, there have been 4 novel de novo mutations identified: 2 nonsense variants, a splice site variant, and a missense variant. Gene expression is the process by which the instructions in our DNA are converted into a functional product, such as a protein. Gene expression is a tightly regulated process that allows a cell to respond to its changing environment. It acts as both an on and off switch to control when proteins are made, and as a volume control that increases or decreases the number of proteins made. The CHD8 gene is highly expressed through the sural nerve with an expression score of 97.06, the ventricular zone at 95.44, cortical plate at 94.28, and the right hemisphere of cerebellum, 94.08. There is also gene expression within the ganglionic eminence, skin of abdomen, anterior pituitary, spleen, Achilles tendon, bone marrow cells, nipple, secondary oocyte, and right uterine tube. Phylogenetics is the study of evolutionary relationships among biological entities, often species, individuals, or genes. The CHD8 human gene, according to the phylogenetic tree, is closest in evolutionary proximity to the gorilla, chimpanzee, and the bonobo. Protein structures carry out a wide variety of organism functions, including DNA replication, transporting molecules, catalyzing metabolic reactions, and providing structural support to cells. Alpha helix and beta pleated sheets are types of the secondary structure of proteins. In the alpha helix protein, a hydrogen bond is formed between the NH group to the CO group of the amino acid. The second essential type of secondary structure of a protein is the beta-pleated sheets of protein. They consist of a various beta strand linked by hydrogen bonds between adjacent strands. Alpha helices are formed like a right-handed spiral, whereas beta-pleated sheets look like an accordion fold. Here is an example of the CHD8 protein, and we can move it around in a 3D way here to see. These are the alpha helices here. And then you can also see some of the beta pleated sheets that are in here. Comparative genomics is a field of biological research in which the genome sequences of different species, human, mouse, or any other organisms from bacteria to chimpanzees, are compared. By comparing the sequences of genomes of different organisms, researchers can understand what, at a molecular level, distinguishes different life forms from one another. Comparative genomics is a powerful tool for studying evolutionary changes among organisms by helping to identify genes that are conserved or common among species, as well as genes that give each organism its unique characteristics. Introns and exons are nucleotide sequences within a gene. Introns, which you can see here as in the example, are removed by RNA splicing as RNA matures, meaning that they are not expressed in the final messenger RNA product. While exons, which here is an example of that, go on to be covalently bonded to one another in order to create mature mRNA. If we observe the CDH gene human comparison to the chimpanzee, there are 31 exons that are shared. Sequence alignment helps to identify regions of resemblance in DNA, RNA, or proteins due to functional, structural, or evolutionary relationships between the sequence. When E, or the expected values, are closer to 0, the sequence alignment carries less error and holds more significance. The CHD8 gene has 0 expected values for Homo sapiens, Pan-Paniscus, and gorilla. Therefore, the CHD8 gene is considered homologous across the expected values. Percent identity measures the number of identical matches in relation to the length of the alignment. The Homo sapiens identity score is 100%, while the Pan-Paniscus and gorilla equally match with a 99% similarity score. Thus, the sequences are very close in terms of evolution. A gap in the sequence alignment means that one or more amino acid has been deleted. In this sequence, Homo sapiens, Pan-Paniscus, and gorillas all have 0% gaps. This means that there are no observed gaps when comparing the three. The gorilla, however, has 1/8,467 versus 0/8,467, which Homo sapiens and Pan-Paniscus share. Hence, the CHD8 gap is closer between Homo sapiens and the Pan-Paniscus. The article, CHD8 mutations increase gliogenesis to enlarge brain size in the non-human primate, was incredibly fascinating. It talks about glial cells, and so as a side note, they are non-neural cell types found within the nervous system, which are typically involved in a range of functions. They provide metabolic support for neurons, they produce myelin, and they play a role in the immune system. The research studied non-human primate models of brain disease to indicate if there were differences in brain pathology. Gliogenesis was detected to correlate with enlargement of primate cerebrum, and CHD8 mutations were thoroughly investigated to identify how these mutations affect the development of the primate brain. The scholarship found an increase in gliogenesis to cause macrocephaly and autism symptoms. In this picture, the scientists examined whether CHD8 mutations caused macrocephaly by closely examining the brain size of the CHD8 mutant monkey against a control. They concluded that the size of the monkey's head, or M3 here on this diagram, was apparently larger than the same aged and sex-controlled monkey. They also performed X-rays and MRIs, which further proved their theory on the connections between gliogenesis and CHD8 mutations. quote, "Our findings suggest that gliogenesis is critical for brain size in primates, and that abnormal gliogenesis may contribute to ASD," end quote. There are 13 transcription factor binding sites for the CHD8 gene. For the purpose of this presentation, I have chosen to focus specifically on the GR alpha site. GR functions as a hormone-dependent transcription factor that regulates the expression of glucocorticoid responsive genes, which probably represents around 3 to 10% of the human genome. The GR alpha form is expressed in most tissues and is the form that binds ligand-causing changes in gene expression. The gene CHD8 encodes the protein chromodomain helicase DNA binding protein, which is a chromatin regulator enzyme that is essential during fetal development. CHD8-related neurodevelopmental disorders with overgrowth are characterized by generalized overgrowth, developmental delay, intellectual disability, autism spectrum disorder, neuropsychiatric issues, and gastrointestinal issues. Most anti-inflammatory effects of steroids are mediated through the glucocorticoid receptor alpha isoform, or GR alpha, which suppresses expression of inflammatory genes through mechanisms. Research suggests that gliogenesis is critical for brain size in primates, and that abnormal gliogenesis may contribute to autism. Biologically speaking, if I had the opportunity to conduct further research related to the CHD8 gene, I would specifically address a connection between gastrointestinal flora with autism, or the brain-gut relationship. I just want to wish you all a wonderful end of your semester, and a big thank you to Dr. Luther for your guidance throughout this semester.